Well, hello, and we are now in Bio 121, switching our attention to this concept of what we call metabolism. And metabolism is the sum of all anabolic and catabolic reactions inside of an organism, regardless of a slime mold, earthworm, I don't know what's outside my window here, some insect flying by the window, makes no difference. And then, of course, there are humans. And your textbook tries to flip and flop between different types of organisms, but at the end of the day, I think it's really important if you're trying to learn this and you think about a way to make it personalized, you're more likely to understand what's happening at the individual cell level. And even though we've sort of given you this idea that cells are used to build tissues, tissues are used to build organs, organs are used to build organ systems, you can begin to understand that to regulate all of this takes a great deal of energy. So up until this point in time, we built a cell, we built some plasma membranes, and then we need to consider, where does that energy actually come from? And if you've already read chapter 6, you know they start where a place I would probably start, and that is, all of the energy that we concern ourselves with in our known existence. This is supposed to be your sun, right? And that sun is undergoing a whole series of hydrogen fusion reactions. So essentially it's a thermonuclear reactor. And what it's doing, of course, is it's releasing frequencies of energy as a result of all of that. And, we are X number of planetoid celestial body structures spinning around this. Oops, I got rid of my sun. Sorry about that. I didn't mean to get rid of the cartoon. Let's go back to yellow. Let's make it a simpler sun now. Okay, let's draw some planetary structures for you here. Let's put Mercury over there, maybe Venus over here, and here's our planet over here. Let me put some dots over here for the remainders of them. But the idea, of course, is as that energy is exiting from this distance, which is like 93 million miles or so, what has to happen is it's going to run into matter. And that matter, of course, is what makes up this celestial body, remembering, of course, we have our own lunar structures spinning around us once every 25 to 28 days sort of thing. All right, so it's spinning around us. Yay. And we're spinning around on top of ourselves. Yay. And then we're spinning around this, right? Which means that any given point in time, some surface area of the Earth will be in contact with some radiant amount of energy that's coming from the sun. And this is how we create our days, this is how we create our seasons, this is how we create what we consider to be time spent traveling around this structure. Just for yucks and giggles, we'll call it 365 days. Now, what's significant about that, of course, is that if life is, of course, built around this concept that you have these individual cells, here's a cell for you here, it means that somehow when we've built this entire structure, we somehow have to make sure that we have enough energy for everything that needs to happen inside of here. And that means we need to be able to give the instructions for this entire unit that might be inside of this entire unit. Or maybe it's this little unit here. Bunny, put a big tail on you, put some big floppy ears on you, a couple of legs. All right. You get the idea that whatever organism it's in, enough energy has to come inside here. And you need to remember when we're going through metabolism that even though your textbook drops you off pretty much in the deep end of the pool here, that somehow materials have to get through both sides of this membrane, and that materials tend to move down their concentration gradients, and that whatever the solute is, remember our universal solvent, which I will change to blue here for you because water characteristically is colored blue. Okay, so if this is diffusion down its concentration gradient, remember osmosis will follow whatever that solute is. So solvent follows solute, and then everything has to happen inside of this environment. And our cartoons inside your textbook make this look really simple. There are some very nice videos inside of this that will help you understand what's actually going on in terms of metabolism inside of living organisms. Let's get rid of all of that. Let's think about what actually is happening. So metabolism refers to the chemical reactions inside of a cell or an organism. What's going to happen, of course, is you're going to change the location of energy as it moves from the sun, collected by photosynthesis, converted into some intermediate that the cells will use, and that intermediate is glucose. And this should explain to you why I've made such a big deal out of this molecule glucose so far. Okay, and what it's going to do, it's going to be stored, and what we do is we use that for cellular respiration. So inside a living organism, you have external respiration, which is gas exchange inside of their respiratory structures, gills in fish, lungs in humans, trees, yada, yada, yada. You get the idea. Okay, but the idea, of course, is that inside an individual cell, in the intercellular respiration actually occurs at the mitochondria. And we break metabolic pathways up into two big fancy words you need to know the definitions of. Anabolic, which are where smaller structures are then added together, and this requires energy, which means you're storing energy inside the bonds where these green balls are actually coming in contact with one another. Now to break them apart, energy will be released, okay? So whatever was stored in between these pieces is now gone, and that energy, I suppose I should draw this for you, because if you think about each of these little things as either glucose or ATP molecules, the free energy that's being released actually looks a lot like radiant energy. Okay, so it's being spent into the space. Okay, and now the sum of these two pathways, okay, are anabolic, catabolic reactions, are what we call in total metabolic reactions. Now, having told you that, when we think about metabolic pathways, there was some prokaryotic ancestor that gave rise to archaebacteria, eubacteria, and the complexity of what's actually happened over time means that a larger organism requires more energy out of the metabolic pathways, but that does not mean prokaryotic structures like eubacteria, archaebacteria, protista, all these structures didn't require energy. And remember, the energy is just going to allow for chemical reactions to occur. Here's an example of one. Here's a glucose molecule plus another glucose molecule gives us now this disaccharide, and that disaccharide now has water, so we know this is a dehydration synthesis reaction. And in the presence of water, when we need to access the energy inside of these molecules to break them down, this catabolic reaction is going to be releasing some energy, but notice what's happened. In the hydrolysis reaction here, we've taken that water molecule, we've added the H here and the OH there. So now you begin to see a little bit of consistency showing up again and again throughout our entire story, that if life has to occur, there has to be some intermediate pattern that we can begin to understand when we look at it quickly. Again, this anabolic reaction is, of course, just dehydration synthesis reactions. The catabolic reaction here is just a hydrolysis reaction, and you can increase the complexity, okay, or decrease the complexity, but in its most simplest, our goal is to get something that looks like glucose available for cells to use. Now, in what ways is photosynthesis anabolic or catabolic, and what evidence can you use to support your answer? And one way to think about this is sort of that story I was telling you. Over the course of a 24-hour period, if you're looking to grow a plant, what do you need? What do you actually end up with? And there's a way to actually think about this, and I want you to go away and think about it. Maybe we'll discuss it later, but what I want you to do, of course, is remember that there's energy coming from the sun, nutrients coming from X location, and then that's all put together to do what? Think about the lawn in your front yard, or the tree that you might see outside your window, or maybe a plant that's growing inside of your house. What happens over time, and how would you use that as evidence to drive, say, a hypothesis that could then be tested, which was done, okay, for multiple times. In fact, that this was answered experimentally, which is why your textbook is asking you to think about it. Now, there are multiple types of energy. We know that there's potential energy and there's kinetic energy, and a way to think about this is the kinetic energy of actually doing your reading, thinking about what I'm talking about while we're lecturing, is energy and action, and that actually releases heat. And believe it or not, while you're thinking, you get more blood towards your brain because the mitochondria inside of your nervous system require more energy, the cells require more energy, so therefore your mitochondria require more resources. After you've eaten a meal and you've had some time to think, you're sitting around, what's happening, of course, is you're storing most of those nutrients in the sense of potential energy, so that when the following day comes along, or the plant grows a little bit in the morning, or whatever, the potential energy is now accessible. This, of course, everybody has potential energy, to do work, but the reality is not everybody converts it into kinetic energy in the appropriate ways. Types of energy. When we're thinking about what this means is how does it all happen? This gets us back to our membranes. And you might remember that we discussed how it is you can create an electrochemical gradient across a membrane, and if these have charges on the outside and there are no charges on the inside, as those materials move, you can actually create an increased amount of kinetic energy that travels between a space. The energy of a chemical electrical gradient across the membrane will allow energy to be generated. In fact, this is what we use in excitable tissues to signal across the membrane. And chemical energy itself, let's see if I can move this out of the way a little bit, is energy stored in chemical bonds, and this would be that hydrolysis reaction that we're looking at over here, so we can actually make these materials available to further catabolize. Now, we're teaching this to you for a reason, right? It's not just because we think it's fun, although it is, If you think about if you own a car and you're driving someplace, all of those former ferns and dinosaurs that we've extracted out of the ground, we've processed, and as you combust, you mix oxygen with this, you add pressure, you add heat, the bonds are broken apart, and that pushes the pistons inside of this race car so it can go around the track in circles and circles. Now, racing cars has never been exciting to me, but the person who's racing it gets a certain thrill, and the people who bet on it get a certain thrill. What I need you to understand is that the potential energy between the bonds of these hydrocarbons are what are driving that reaction. And there was this dude, his name was William Gibbs, Henry Gibbs, I don't remember, but his last name is Gibbs. And he actually has a lab up in New Haven. And he's part of the chemistry department, right? And we're talking 100 years ago now and change, where what he did is he asked, well, if there's energy that was required for reactions to go forward, it has to be accessible, and it is accessible in the sense of what we call free energy inside of any bioenergetic system. And what happens, of course, is G, the Gibbs free energy, is the total amount of energy available to do work, the usable energy that is available to get that work done. And what he did is he came up with this experiment, this description of free energy that's available, where he's defined that the delta H is the change in the total energy of a system, subtracted by the temperature in Kelvin degrees, and the delta S is the change in the entropy. It's either lost to disorder or it's stable to disorder, in which case you can then begin to measure the amount of free energy that was required. And for normal chemical reactions to go forward, the delta G here tends to be a negative value. For you to add energy into the system, the delta G becomes a positive value. And the idea is pretty simple. If you have, say, a ball rolling down a hill, gravity is assisting it and it requires no energy from the ball, so therefore the energy is taken away. To get the ball to roll up the hill, you have to add energy to the G, and therefore it would be a positive reaction. Now, it's not the best of examples, and in fact, we can't actually get into a lab to demonstrate how specific reactivities using free energy available to them change the amount of disorder inside an environment that can be measured as a temperature change, which is what Gibbs did to demonstrate that this actually was a significant sort of observation. How we use it, though. Exergonic reactions are where heat will be released into the energy system because less free energy the products of these reactions will have less free energy than their substrates, meaning that at the end, less potential energy can be measured than when we began. And this is just our A plus B. Our delta G is minus 0, meaning it's below 0, and therefore the energy that's released into the system can be determined here. And notice what's happened is the final products, the A, B over here. What this would mean is that there's less potential energy inside of those bonds. If you see a tracing that looks like this, it means that this is an exergonic type reaction. And exergonic type reactions are spontaneous reactions, and they can occur without the addition of energy. And inside the lab, normally inside a chemistry class at this point in time, somebody would pull out naturally occurring magnesium, which is relatively labile, and what happens if you hit it in the presence of air, oxygen, which is at 18% or so, that's sufficient for it to release light energy. Which means the magnesium is interacting with the oxygen inside that environment and breaking it down. So now, why do we actually care about this? Well, because that if we need to look at an endergonic reaction, those that actually store energy inside of the bonds, here are reactants A plus B. We store energy inside of those bonds. Notice that the products now, AB, over here, would, in fact, have more potential energy. And therefore, that endergonic reaction inside of a lab, what you would feel is, you would actually feel the reaction vessel become cold. That's an important observation. It means what it's doing is it's storing energy inside of those bonds. The bonds are found inside the products. And because of this, what it also means is that Mother Nature has found some way to create a system where the sum total of products that are exergonic and endergonic, even though they may not be at equilibrium, they're approaching some form of equilibrium. And we know that over the course of time, that even then, the entropy of the universe will be approaching zero. So even though we're producing potential energy inside of these products over the course of hundreds of billions of years, that energy will dissipate into space. So now, if we were to look at this, which one of these are exergonic? Which ones are actually storing energy? Okay, or exergonic would be releasing energy, right? And that would be for us to take something apart. So our catabolic reaction here, this would actually release energy into space. Because we're actually making a more concentrated product here, the potential energy, if we're thinking about what's going on, we would actually see that, well, maybe we have to think about this and think about what Mother Nature is hoping to do here. So why do we care about this? Okay, in this last couple of minutes, we need to introduce this concept of what energy of activation is doing. And this is why catalysts and enzymes are very important for you to understand. Catalysts tend to be metals in the naturally occurring elements of, our planet. And what happens is they stabilize the reactants of a chemical reaction. Enzymes are proteins that act as catalysts, and they are not destroyed inside the process of the chemical reaction, but because they're a protein, it means that they're built specifically for that one unique reaction. Catalysts on the whole are metal because they're metal, they have a unique shape, and they may drive a reaction, but it may not necessarily be specific. It just might be opportunistic. Here's the important point, though. Activation energy is the energy that's required for a reaction to proceed. You're sitting there, you're doing your homework. Really what's going to motivate you to actually get your homework done is your fear of your grade. So that's your catalyst to lower the energy of activation, right? You say, oh, I'll wait till tomorrow, but the test is tomorrow and you actually need to read tonight. So here's the way you would approach that. And believe it or not, even though it's probably a really bad example to say that energy of activation to actually get the products here have less potential energy, the way to think about that would be that what you're doing is you're consolidating knowledge and you're reducing the amount of entropy of knowledge inside of your brain. So that's a pretty good place to stop. We'll pick up here in a couple of minutes. Again, I'm trying to keep these presentations a little bit on the shorter side to make sure you actually pay attention to them. Be safe.